So, for this worksheet, we're going to talk a little bit about Sanger sequencing. And to kind of get a good grasp of Sanger sequencing, it's good to kind of renew, refresh our understanding of PCR. So, we're going to start off with making some PCR primers for a target gene. And then we're going to talk about how we can use those to sequence that gene. So, let's first just kind of review what PCR is. So, remember with PCR, remember we were talking chapter 12 and we're doing DNA replication, and we separate our two strands of DNA at our origins of replication. We start using each side of the DNA molecule as a template to make the other side, and we do that across the whole genome and copy the whole genome. We're going to be doing that in the test tube in a very targeted way, because remember when we open up at our origin of replication that primase is going to be putting down primers, and the reason for that is so that DNA polymerase needs a 3' hydroxyl to be able to synthesize DNA. So what we're going to do is we're going to do the job of primase, and we're going to design our own primers for exactly what we want to make copies of in the genome. Now, so when we do PCR, we're going to need some template DNA. Often this is going to be genomic DNA from, say, people or whatever, some tissue, some source of DNA. Can be any source of DNA. We're going to have our polymerase. It's a Taq polymerase. We've talked about this being a thermal stable one that can withstand high heat. DNTPs, so all of our A's, T's, C's, and G's, and then our primers, and then we're going to run this through a series of reactions, or a cycle it through temperature changes. These temperature changes are, remember, we heat it up to 95 degrees, then we let it, and that's going to disrupt all the hydrogen bonds. Everything becomes single-stranded at that point. And then we're going to start cooling the reaction, which is in this little test tube. And as we cool, we'll hit a certain temperature that the hydrogen bonds can start to reform again. Now you got to imagine that say the genomic DNA are these really massive, enormous chromosomes, and as we cool the temperature, they could anneal back together, meaning reform their hydrogen bonds, but it takes a little while because they're such huge molecules. They got to kind of find their complementary sequences again. Meanwhile, we're going to have put tons and tons of our primer in there, and this is a little tiny 20 nucleotide sequence. It's going to stick onto the target spot quick and easy, and then we heat the solution up to 72 degrees Celsius, which is the optimal temperature for DNA polymerase or the Taq polymerase to elongate. And so remember that when we're doing this, that we have so here's our template DNA. We're going to heat up the DNA, and that's going to cause it to be single-stranded. And then our primers, notice the way our primers are. We're having the 3' hydroxyls are facing each other. We have 3' over there, 5' over there, we have 5' over here, and 3' over there. Okay. And so what that does is where our 3' hydroxyl is, that's the direction I can synthesize. Where our 3' hydroxyl is, that's the direction we can synthesize. If this top strand over here, I had my 3' over here, I'm going to synthesize in that direction, and that's not where I want to go. Okay, so what we need to do is we need to design primers that are facing each other, meaning the 3' hydroxyls are facing each other. Okay, so we go through this reaction over and over and over again. Every time we denature, the new strands of DNA that we made become template for the next strand of DNA. Etc. Okay, so now let's worksheet. So imagine you come across this population that has superhuman speed. Okay, and so you get a bunch of people in this population, both normal speeded individuals and super fast people, and they give you all DNA samples so you can make some comparisons and try to find the gene responsible for the super speed, and maybe be able to identify the mutations. So now you go through, you do some linkage analysis, and you identify the region of the genome. You go and look, and you find a gene, and you're going to say, this gene might be responsible for super fast speed because it's linked to the trait, and so we're going to call this gene our run gene. Okay, so that's the gene we have here, and so in bold is the open reading frame. And the first task we have to do is design primers for this. Now, in this example, we're going to do 6 nucleotide long primers. Typically in PCR, we're going to be doing more like 20 nucleotide primers, just because the longer they are, the more specific they are, they're only going to land at one spot. But for illustration purposes, we're going to do 6 nucleotides. And what we want to amplify is just the bold region. Okay, so in order to do that, I wanted to have my primers start at this A and end at that A. Okay, I don't want them going into either of these sequences on the side. All right, so to do this, the easiest way that I would recommend is to write out your the other strand of the DNA. And so remember, this is going to be a double-stranded molecule. So I have 5' on the top. That means I need 3' over here. And then over on this side, on the right, I have 3'. So on the other strand, I need 5'. Okay. Now, then the sequence of the DNA is going to be complementary. So where I have a G, I put a C. Where I have an A, I put a T, T, A, etc. And I'll fill that in. Okay, so I filled in that sequence. And so where I want to amplify is I want from there to there is what I want to amplify. All right. So now remember what we want our primers to be doing. We want our primers 5' to 3', 5' to 3'. We want our 3' hydroxyls facing in each other. So when we go 5 to 3, 5 to 3, they're facing towards each other. So this actually kind of just occurred to me as I was working through this worksheet, that probably the easiest way to kind of identify this is, so on the left, this is going to be our left primer. This is going to be our right primer. Okay, on the left, we need the sequence of DNA that starts at the 5 and goes to the 3. So the top strand starts at the 5 and goes to the 3. And so that becomes our first primer. Okay. Because remember, what's going to happen is we're going to heat up our DNA, which will cause it to be single-stranded like we see here. And then as we cool, this sequence, A, T, G, A, T, T, 5', 3', is going to bind to this location. So now DNA polymerase can come here and synthesize DNA going in that direction. All right, so now the right primer, we need to have 3' on the left and 5' on the right. Okay, in which case that is the bottom strand. So we have 3 over on the left, 5 on the right. So that means that the first 6 nucleotides of the bold sequence, okay, remember, make sure you're designing for what you're designing the primers for where. You don't want to design the primers over here because you're targeting just the open reading frame. So that will become your second primer. So as you go into your annealing step, it can hybridize right here. And then there's your 3' hydroxyl, 5', and so you can synthesize going that way. All right, so our left primer is 5' ATG, ATT, 3'. Our right primer, by convention, we write things in 5' to 3' direction, so we're going to start at the 5', TTA, GTG, TTA, GTG, 3'. All right. So now, what is the full length of our sequencing product? The full length of the PCR product, or what's the sequence of the full length of the product? So this is just to kind of illustrate that what we're capturing is where our primers start and end and everything in between. And so we can just write that sequence out as 5' and 5' ATG. Okay. So now, that would be what, when we amplify and we start with genomic DNA, the whole genome, we run our PCR reaction, and what we get is millions and millions and millions of copies of this sequence. So remember, when we're doing this, that this sequence is actually double-stranded. Okay? And so we can kind of just fill this in because it will be helpful in a minute. Okay. So now let's look at, so that's what is in our PCR reaction. So the context of this is how this would be useful is, so say I brought up in class about, say, the BRCA1 or BRCA2 genes that we know increase the risk of cancer susceptibility in women. That we know there are certain mutations that if you have one of these mutations in your BRCA1 or your BRCA2 gene, we can tell you that you are at an increased risk. So what you would do is you take that patient that came to you and said, I have a history of breast cancer in my family. I'm wondering if I have an increased susceptibility. And so you would maybe take a little swab of the inside of her mouth to get some DNA, PCR the BRCA1 gene or the BRCA2 gene to amplify it. And then you take that PCR product and put it into a Sanger sequencing reaction to find out what her sequence is for that particular gene. Okay, so that's what we're going to do now. So in number 3, you're going to run the Sanger sequencing reaction on this PCR product from a homozygous normal individual. Okay, so this isn't a fast runner. This is just a normal person. Using the left primer for the run gene. In one of the tubes, we're going to put our DNTPs, okay, in molar excess. That means we're putting a whole ton of all of our normal nucleotides and a little tiny amount of our DDGTP. So this is the dideoxy guanine. And so remember what's going on in our Sanger sequencing reaction. The Sanger sequencing reaction is essentially the PCR reaction. We're putting in template DNA. In this case, we're putting in PCR product rather than genomic DNA. We have our Taq polymerase in there, just like a PCR. We have our DNTPs, just like a PCR. And then we're going to do our dideoxynucleotides. And so remember, dideoxynucleotides are these that are lacking that 3' hydroxyl. And so when we incorporate one of those, remember these are in low abundance, so most of the time we're not going to incorporate one, but sometimes we will. When we incorporate it, the strand can't grow anymore, and we just have a piece of DNA that big, the primer up until that nucleotide that got incorporated. And so, well, and traditionally the way it used to be done, that we've talked about, is we do 4 different tubes where everything's all the same template primer, where we're only using 1 primer. The reason we're only using 1 primer is because if I was doing 2 primers, I would be detecting the sequence for each side, and that would confuse things, because if I was coming from this way, I'd have a first nucleotide of a G and the first nucleotide of an A, and I'd get 2 bands. Okay, so I'm only going to do 1 primer at a time. Split it all up into the 4 different tubes where I put a little bit of each dideoxynucleotide. Okay, and so as I run this reaction, just like a PCR reaction, most of the time on most templates, I'm going to go all the way through. But occasionally, let's say in this A tube, when I get to the first A, so G and then an A, it's going to get a dideoxynucleotide, in which case it just truncates and I just stop. Now that's still in a tube, and those are going to start accumulating. And then this size fragment is going to start accumulating as well. And this size fragment is going to start accumulating as well. And so we're going to just keep on accumulating these different fragments in each tube that's representative of the primer plus however many nucleotides ending in that particular nucleotide. Right? And so when we, so here, and this is going to be looking at just the G tube, the tube with just the GTP. You visualize this product on a gel. How many pieces of DNA would you get? Okay, so there's really kind of 2 ways that this you can answer that question. So what we're doing is we're putting in this primer, the left primer, which means the primer is going to land right here. So I'm making this top strand, okay, what is I'm going to be visualizing on my Sanger sequencing gel. Okay, so the very first band is going to be in the A lane. The second band is going to be in the C lane. The third band is going to be in the G lane. So in this case, I'm using DDGTP. So at every spot that I get a G, in this sequence, I'm going to get a fragment. That is my primer plus that many nucleotides. My primer plus that many nucleotides. My primer plus that many nucleotides. And technically, I will get a product that is my primer plus the full length. So you really actually are getting, while you have 3 short products, you technically will have 4 products because you get your full length one, though we're not going to be concerned with that one because that one's, so we're going to get 3 short, and so how many pieces of DNA would we have? 3 short with G terminating, and then our full length. The full length is the whole thing. Okay. The ones we're interested in are the ones that terminate with the G. Okay. So what would be the sequence of the fragments of DNA made in this reaction? Well, we'd have our primer. We won't actually see that in the gel, so we don't see those letters. We don't count the letters. The very first letters that we're going to see is that first nucleotide. Okay, so in this case, remember we're talking DDGTP, so we're going to have ACG. We're also going to have fragments that are our primer plus ACG down to this second G, TCG. And we're also going to have fragments that are 5' and then go all the way down to that third G. ACG, TCG, ATAC, TCG. Okay. And then full length ones, but those aren't the ones we're interested in. In the full length ones, the reason you're not going to be interested in them is because in the way we do it now, remember how we talked about how we put all of the nucleotides into your tube and they're all fluorescently labeled, and then we run them through a laser. And so the full length ones are the ones that never incorporate a dideoxynucleotide, so you'd never actually visualize them. Okay, so what we're interested in are all of the truncated ones, all the ones that got terminated, and then what we can do is then at the lowest band on our gel, that's going to be primer plus 1 nucleotide. The next lowest band is primer plus 2 nucleotides. That one's in the A, primer, the green, plus 1, 2. Okay. So we can just read our sequence. G, A, T, T, C, G, A, G, C, T, G, A. Okay, from the 5' to 3' way. Okay. Now, so when we're looking at our worksheet here, we now need to draw this sequence out on the gel of what we would get. And so for the sake of making it easy, I'm going to write that sequence up above, right up here. Okay, so now this would be the sequence that we're making. So remember our primer is the first six nucleotides. So we're not going to see those sequences on the gel. That's an important thing to remember. That is our primer. So the first small fragment we get is going to be here. Okay. Take that back. The first fragment we're going to have is an A. Okay, and then we're going to go C, and we're going to go G. So that's 1, that's 2, that's 3, and that's 4, and then that's going to be the 5th position. Okay, and so the way we fill out this gel, we're going to have 1, 2, 3, 4, and 5, and 6, and 7, and 8, and 9, and 10, and 11. So we're going to get through the first 11 positions. So primer plus 1 nucleotide is an A. Primer plus 2 nucleotides is a C. Primer plus 3 nucleotides is a G. Primer plus 4 nucleotides is a T. Primer plus 5 nucleotides is a G. back. Instead of writing complements, we don't want to write complements. I'm not writing, this is not the complement in red. That is the actual sequence. And so what we want to do is then go to C. And then next is a G. Next is an A. Next is a T. Next is an A. So that is the 8th nucleotide, and then the 9th nucleotide is an A. OTA and C. And back to T. Okay. And so now, so that's what the gel would look like. So now how many and what types of genetic changes, or single nucleotide polymorphisms, SNPs, so single letters that change, does the superfast human have? Okay, so this is the fast person. This is the normal person. So if we look, okay, we got A and A. We have a C and a C. We have a G and a G. And here in lane 4, the 4th one, we have a T, which we have there, but we also have an A. Okay. So in this one, we have a T to an A mutation. And then after that, we go to a C and a C, and a G and a G, an A and a T and that's not a T, so we have a T to a C. And then the 9th, we have an A to an A, and a C to a C, and then a T to a T. So we have 2 different mutations here. Okay. We have a T to a C, and a T to an A. So this is equal to, remember, cut the pyramid. These are both pyrimidines. Transition, and that is equal to a transversion. And you probably noticed something funky about that right gel. That's what the last question is. Why are there 2 bands in the right gel of the 4th position? So right here, where this arrow is. And the reason for that, what do you think? This person is a heterozygote for an A and a T. Okay. And so with that, that sums up that paper, and that's how Sanger sequencing works. Have a great day, everybody.